Okay, thank you for the introduction. Good afternoon, everyone. I promise to stick to time. There are two sources of urine oxalate, endogenous oxalate synthesis and dietary oxalate absorption. Yeah, so there's two sources, endogenous oxalate synthesis and dietary oxalate absorption. So I'll touch on some of the knowledge gaps around both of these processes. We've only just begun to look at endogenous oxalate synthesis in healthy volunteers. We do this using very low oxalate diets to sort of limit the interference from dietary oxalate. And we've also been using continuous intravenous infusions of carbon-13 oxalate. And the figure here shows daily oxalate synthesis rates in 9 individuals. And what really struck us was this considerable variability between the individuals. It ranged anywhere from 11 milligrams to up to 26 milligrams. And so a major knowledge gap is really, we don't know what factors drive this variability and influence endogenous oxalate synthesis. We did look at sex as a biological variable. It does appear that females synthesize less oxalate than males. Females here were at 15 milligrams and males were 7 milligrams higher. But there are, probably other factors that are involved. Obesity and type 2 diabetes are associated with increased urinary oxalate excretion. So it's possible that these conditions have heightened endogenous oxalate synthesis. And we haven't yet embarked on looking at endogenous synthesis rates in calcium oxalate kidney stone formers. And this is certainly something that we need to focus on in the future. There are 2 precursors that are involved in the synthesis of oxalate that we know of, glyoxylate and vitamin C. However, the regulation of these pathways is still very unclear. We know lactate dehydrogenase oxidizes glyoxylate to oxalate, but the specific factors that influence LDH-mediated oxalate synthesis is still poorly understood. There are probably other dehydrogenases as well that are involved in this reaction. There has been some interest in xanthine dehydrogenase, for example, that may catalyze this reaction as well. We know very little about vitamin C breakdown to oxalate. It's known to be a non-enzymatic process, but no studies have really been done to understand this process in tissues in vivo. Another knowledge gap is we really don't know what the contribution of all the tissues in the body is to oxalate synthesis. The liver is believed to be the largest source, but all the ingredients and machinery for oxalate synthesis are also found in other tissues. So this is another area that should be looked at. And we still don't know what the contribution of both glyoxylate and vitamin C is to oxalate in kidney stone formers and whether it's any different from non-stone formers. Moving to dietary oxalate and its contribution to urinary oxalate excretion. A lot of work's been done on the important role of dietary calcium. We know this limits dietary oxalate absorption and therefore urinary oxalate excretion. What I'm showing here is a culmination of studies we've done over the last 20 years. And in the black squares, this shows a diet fixed at 1,000 milligrams of calcium and varying from 50 milligrams to 750 milligrams of dietary oxalate, which is the normal range of intake. And from the line of best fit, we can see that you only absorb about 3% of dietary oxalate when taking in a recommended amount of calcium of 1,000 milligrams. But as shown by the triangle, when you decrease calcium, you get this profound increase in urinary oxalate excretion. So we have a relatively good understanding of dietary calcium and its importance, but there are a number of knowledge gaps. I've only highlighted 2 here. A major one is we still have a poor understanding of gut oxalate transport. Where is oxalate absorbed? There was some early evidence that suggests some is absorbed in the stomach, but that hasn't been followed up. We also don't know what the relative role of paracellular versus transcellular absorption is. And we don't really know what the main transporters are throughout the gut that are involved in this process. There's also been a lack of controlled dietary studies in kidney stone formers, calcium oxalate kidney stone formers. So our understanding of dietary oxalate handling in this cohort is still poor. I just wanted to touch on enteric hyperoxaluria. This is a set of malabsorptive conditions that lead to increased dietary oxalate absorption and, as a result, hyperoxaluria. I've listed some common causes here: bypass surgery, inflammatory bowel disease. And a lot of the research now is focused on developing therapies that increase gut oxalate degradation. And Alina Pharmaceuticals, some years ago now, completed an enteric hyperoxaluria clinical trial using an oxalate-degrading enzyme therapy. And the figure shows that in their treatment group, there was a 23% reduction in 24-hour urine oxalate excretion with their enzyme therapy, and a 10% in the placebo. So the net effect was only around 13%. It's still somewhat unclear why the trial failed, but I wanted to bring this up because it really just highlights our very poor understanding of the sources of urine oxalate and dietary oxalate handling in stone formers with enteric hyperoxaluria. There's a lot of interest in oxalate-degrading gut microbes. Only one study has looked at oxalate-degrading function in healthy volunteers, and this is what this figure shows. In the study, stool oxalate was measured on fixed dietary oxalate diets. There were two cohorts, one that was not colonized and another that was colonized with a specialist oxalate-degrading bacterium called Oxalobacter formigenes. And what it highlighted is, firstly, how central this specialist oxalate-degrading organism is to efficient oxalate degradation in the gut, the lower white bars here. But the other thing the study highlighted was at the higher oxalate intakes, you're getting appreciable oxalate degradation from other microbes. So at the higher 750 milligram intake, for example, about 60% of the dietary oxalate was recovered in stool, suggesting microbes were involved in about 40% of the breakdown of oxalate. We have numerous knowledge gaps. I've only listed 3 here. A large one is we don't really understand the factors that influence colonization of the gut with these oxalate-degrading bacteria. We know Oxalobacter formigenes is sensitive to commonly prescribed antibiotics, but the sensitivity of these other oxalate degraders to antibiotics hasn't been defined. There's probably, host-derived factors, microbial-derived factors that are also influencing colonization of these bacteria. Controlled dietary studies haven't been done to look at oxalate-degrading capacity of the gut microbiome in stone formers. So this is work that still needs to be done to see if there's a difference between non-stone formers and stone formers. And most importantly, it's still somewhat unclear what the impact of these bacteria in the gut have on urinary oxalate excretion. Just to end with I wanted to give some final thoughts on potential future research that I think would be better served with a more collaborative, multi-center approach. As I mentioned before, we don't really understand the contribution of endogenous oxalate synthesis to urinary oxalate excretion in kidney stone formers. This can be examined using very low oxalate diets. I think it's particularly relevant to look at this now because there is an approved therapy to reduce endogenous oxalate synthesis and numerous others that are in the research pipeline. I think we should increase our efforts to sequence genes involved in oxalate synthesis, as this may identify at-risk cohorts. And we need to perform controlled diet studies to better understand the sources of urinary oxalate and dietary oxalate handling in kidney stone formers with enteric hyperoxaluria. I think the results from these types of studies will really help us design more rigorous clinical trials to test these emerging oxalate degrading therapies. And a lot more work needs to be done looking at these gut oxalate degrading microorganisms and their impact on urinary oxalate excretion. And a novel area of research as well, which these controlled diet studies will help with, is sort of increasing our understanding on whether bacterial metabolites can impact either or both endogenous synthesis or dietary oxalate handling. So thank you all. Thank you, Chris, and to the committee for including me in the program. And happy to take questions later.